Welcome to the Good Chemistry Podcast. My guest today is Dr. Jonathan Page. John is a botanist and entrepreneur who has deep expertise in the cannabis plant. John has a PhD in botany from the University of British Columbia and helped lead the team that published the first cannabis genome sequence back in 2011. In addition to a long research career studying cannabis and plant chemistry, John is also a successful entrepreneur. He founded and was CEO of Anandia Labs, which he started in Canada in 2013, and later sold to Aurora Cannabis in 2018 for around $100 million. John served as Chief Science Officer for Aurora until just recently. And John and I spoke about all things cannabis science, ranging from the botany and natural history of the cannabis plant and what the plant does, the chemistry and biology of cannabis and its psychoactive effects, and whether or not different types of cannabis can cause different types of effects and how that relates to its chemistry. John also shared some amazing stories about transitioning from scientific research to starting, building, and selling a cannabis company, and shared his thoughts on what's next for the industry. If you enjoy this content, please consider liking, sharing, or subscribing. And with that, here's my conversation with Dr. Jonathan Page. John Page, thanks for joining me.
Hey, thanks for having me on, Nick.
How are you doing, and where are you calling in from?
I'm doing pretty well. I mean, I say that with all the concerns around COVID that we're all experiencing. And I'm actually in my dining room, which sort of doubles sometimes as my home office on the west side of Vancouver in British Columbia, Canada.
Excellent. And can you just start out by saying a little bit about your background in science? So what are your degrees in, and what are your scientific credentials, basically?
Yeah, so I have a Bachelor of Science degree, an honors degree in biology from the University of British Columbia here in Vancouver. And I also have a PhD in botany from the same institution, from UBC. And I did those pretty much back to back. Took a year and a half off between an undergrad and a PhD. And then I left Canada as a postdoctoral scientist, and I went to Germany, first to the University of Munich in Munich to do, my PhD was essentially on phytochemistry, so identifying new molecules from plants that have, in this case, they had antibiotic properties. Mm-hmm. And I went to Munich, sort of change things up and really get into the world of biosynthesis. So, how plants make secondary metabolites or specialized metabolites. And so I was studying alkaloid biosynthesis in Munich. And then the lab I was with moved to a new institute in a city in the sort of middle of Germany called Halle. It's actually a really old historic site or a historic center, but also an academic center going back hundreds of years. And I worked at a Leibniz Institute of Plant Biochemistry there, also doing biosynthesis work. And actually, I went to Germany as a postdoc thinking I would spend a couple of years there. I was really there for almost 4.5 years or something. And sort of moved from being a postdoc to a group leader, having my own small research team. And then I took a job back in Canada with the National Research Council, or NRC, in a prairie city called Saskatoon. And I worked there. The title was research officer. I worked there for a decade, 2003 to 2013, doing pretty much that, using biochemical and molecular tools to try to understand how plants made specialized metabolites. So that was kind of, both academic training and then into the job world for me.
Mm-hmm. And then how did you get into cannabis? When did that enter the picture for you?
Yeah, there's two parts to that story. Where I grew up, so I'm here today in Vancouver, but I grew up on Vancouver Island I was born in Victoria, and I lived in a sort of smaller community on the east coast of Vancouver Island called Courtenay. And cannabis was pretty much present, let's say, in the environment or the culture and in places like that. So, there was lots of cannabis around. So I was definitely familiar with it, both as a high school student and a university student. But I wasn't formally working on it. I think everyone who was interested in plant chemistry probably thought a little bit about cannabis and THC, no matter if they studied it or not. But the encounter with cannabis on the academic or research side was kind of fortuitous. So I mentioned I finished my PhD. This was sort of like Christmas 1997, and I had NSERC. So that's sort of the NSF, the big federal funding agency for science in Canada. I had an NSERC postdoctoral fellowship that I had to kind of take up quickly, or I would have lapsed or something. So I moved to this lab in Munich, Germany, at the university there, whose main focus was alkaloid biosynthesis. And so their lead, his name was Meinhard Zenk, he had studied all sorts of alkaloids, but in particular morphin alkaloids from opium poppy. So this would be, morphine, codeine, thebaine, the big alkaloids for which that plant is known, and obviously the source of lots of medical value, but also the crisis of opiate addiction in our society. So that plant was intensively studied there, and to do that work, they had the lab there. The professors had a federal drug license from the German government. It was, I think the body is called the Bundesopiumstelle, which is sort of the licensing agency. So they had opium poppy growing in very secure and controlled system up on the roof of this university building, which was like about 3 blocks from the main train station in Munich. And there was a couple floors of labs and then this kind of grow area. But because they had that federal drug license, they were also, and that allowed them to grow opium poppy, but also possess standards for things like morphine, mm-hmm. which they needed for analytical purposes. But they had, a couple of years before I arrived, got interested in cannabinoid production and using some of the tools of they were sort of feeding stable isotope labeled precursors and using NMR to kind of dissect out how carbon flux moved through metabolic pathways. It was a kind of a cool technique at the time. They had applied that technique to cannabinoids, which meant they were growing cannabis as was permitted by their license. So I started a project trying to actually clone an enzyme of alkaloid production, which was involved in making alkaloids like berberine, which is this yellow, almost fluorescent alkaloid that's found in some species of berberis. And actually around where we are in Vancouver and Seattle, it's found in Oregon grape. If you look at the roots or the rhizomes of Oregon grape or Mahonia, there's a bright yellow color to that. So those alkaloids were interesting for medical purposes as well. And I was trying to clone an enzyme involved in the production of those alkaloids. The enzyme is called S-tetrahydroprotoberberin oxidase, or STOX. And I spent about 6 or 7 months sort of learning molecular biology as I tried to clone this thing. And it didn't work. I mean, maybe it was longer than 7 or 8 months, but by sometime in my first year in Munich, I was not making the kind of progress that I wanted to. And I really started to think about other projects and being knowledgeable about cannabis and its importance, and then spending some time on the textbook biochemistry, which really revealed cannabinoid production was pretty much unknown, at least the enzymes that were doing that. I talked to my postdoctoral supervisor, and I had come with my own fellowship money, so I had a bit more freedom. And she was like, "Yeah, absolutely." And so I started, grinding up cannabis and looking, like isolating trichomes and all that kind of stuff, which we could talk about more. But it was kind of like I just happened to be in the right place at the right time with a lab that had probably one of the few drug licenses, even in Germany, that could grow plants like opium, poppy, and cannabis. And I jumped on that opportunity.
So you never really at the beginning intended to study cannabis. You were just studying this other project, and it happened to be in this place that had these licenses. And then your main project basically failed, I think, is what you said. And so you had to shift to something else. And so it was this fortuitous opportunity.
I guess I wouldn't say failed. It kind of faltered. It was just kind of, you come from a research career as well. I mean, projects just bog down and don't move forward. And I would also say that my career in plant biochemistry was generally guided by the fact that I was interested in plants and molecules from plants that had relevance to human culture. And sometimes that was more related to their medical properties, but certainly I spent a bunch of time as an undergrad being totally fascinated by the sort of chemical side of ethnobotany. I mean, I kind of idolized guys like Wade Davis and read all the Richard Evans Schultes books about those days of yahe or ayahuasca. And so I was, very aware of the sort of mediation between plants, or the role that molecules from plants played in human culture. So it wasn't like fortuitous to end up in a lab where cannabis was able to be grown. But I think the sort of field had been sort of prepared for me to be really excited about working on cannabis as the focus.
So before we jump into cannabis, cannabis has THC inside of it. You just mentioned, poppies that have things like morphine. Plants have antibiotics and all sorts of stuff. Why do plants make all of these drugs that happen to be useful to humans in the first place? Can you talk a little bit about the ecology of why that's true?
Well, plants are, amazing chemists. And I think it's a truism that you hear people who teach in this area talk about. I mean, they're sessile. So plants sit there, right? And you just have to, look at the trees outside or something, and you see they're not moving. And so, herbivores and insects and even pathogens that sort of blow in with spores on the wind, they arrive at that plant, and they may be inflicting damage by, say, eating the roots or leaves, and so plants, they have no nervous system. They don't, other than things like Venus flytraps that actually move and trap insects.
They can't run away.
They can't run away. And their world then is very much, their ability to kind of interact with their environment is very much related to their chemistry. And I mean, interact with their environment, like deal with biotic, so, animal stresses, for example. So, they produce many, many chemicals as defenses. Some of those, not every specialized molecule that we find, specialized metabolite we find in plants necessarily has a known function. And, we're still trying to figure out what cannabinoids do. But in general, they're there for defensive purposes, sometimes for more signaling purposes, to signal with other plants or other organisms. Sometimes it's about like UV protection. So things like flavonoids in leaves are probably not there for, defend against a herbivore, but they, making sure that the UV light doesn't damage the plant. But I think things like morphine and cannabinoids, I guess it's nice to think that the plant sort of designed these molecules because mammals or other animals had receptors that those molecules interacted with, and that would therefore provide some sort of defensive mechanism. And you could sort of think that, for example, morphine in poppy, that where opium poppy makes alkaloids like morphine and codeine is in the capsule. That's like the sort of fleshy sort of almost fruit around the seed production. Mm-hmm. And so this is where the plant has invested all its sort of ecological future in the seeds to be the next generation. And opium poppy is an annual, so it lives and dies in one season. So those seeds have to survive. And so when an animal comes along and eats the seed capsule, there's no seeds left. So the evolutionary pressure to protect those seeds is really, really strong. And so there's leticifers. These are little channels that contain latex, kind of milky sap that house the alkaloids in there. And so you could see like an insect would come along and start gnawing its way in to get those seeds. And the seeds are actually quite rich in fat. Opium or poppy seeds are kind of tasty, as humans know as well. And so you could sort of see having things like morphine and codeine to ward away insects or maybe mammals that came along and tried to chew their way into that capsule. But I'm not sure we can even really say that. I mean, it could be that they just provide a real bitterness to and it's just kind of accidental that they kind of interact with our receptors. Or, the latex itself, like, exudes out and gets all sort of sticky and gummy as it gets exposed to air. Maybe that's just a physical aspect to preventing that. So, I think there's been, lots of ideas around coevolution of drug plants and humans, and this idea that, cannabis evolved to produce THC or other cannabinoids because they interact with our receptors, or opium poppy evolved. I'm going to say, in the case of alkaloids, maybe, but with cannabinoids, I don't think that's the case. I think it's just kind of accidental.
Mm-hmm. Yeah, it does seem to be a common theme that when a plant is making drugs of some kind that we subsequently use for our own purposes, they're pretty much always bitter tasting. They don't taste good. It does look a lot like the plant is just making stuff to make itself unpalatable.
Right. Yeah, I think there's not too many human consumers of cannabis who would go into a grow room, at peak flowering and start chewing on that. But that stuff is going to taste pretty nasty.
So what about cannabis, the plant? What kind of plant is it? What's it related to? And where does it come from originally?
So cannabis is an annual plant. And I mentioned that means it grows and dies in a year. In general, though, you can get in more tropical locations kind of almost perennialized cannabis. And in cannabis, inside, greenhouses and grow sites can also live for a long time. But so it's an annual plant. It's a member of the Cannabaceae family. So that's a plant family that's quite small, contains cannabis and humulus. So humulus is the hop plant, the plant that's grown to be used for making or flavoring beer. It contributes the bitter flavor to beer. And just as an aside, I never, and I didn't mention this before, but when I was in Germany and I was working on cannabis, our institute, so I started in Munich and I moved to a city called Halle to join another institute. And then the institute at Halle recruited a new research group from the Netherlands, and they came in and we had a kind of research day where we talked about all our work that we were doing. And I presented a talk on cannabis and cannabinoid biosynthesis, and another researcher who was just going to move with this new group. His name is Fred Stevens, and he's at Oregon State University now. He came up to me and said, "Hey, you're working on cannabis. You should be working on humulus too, because the hop plant is equally fascinating from the chemistry standpoint." And he had done a lot of work on the chemistry of metabolites and hops. And so we started collaborating. So I worked in parallel on hops and cannabis for a while. But so the cannabis plant, it's a Cannabaceae. Its close relative is humulus. It's dioecious, which is a Latin term that I think it translates in English to two houses. Really, what it means is there's a male plant and a female plant. So the sexes are on separate plants. I guess the other thing, a few sort of features of cannabis that make it interesting. Obviously, it produces cannabinoids. There are a few other plants in the world that produce cannabinoid-like compounds, but cannabis is really the only one that produces this plethora of cannabinoids like THC and CBD. It produces bast fiber. So, cannabis is used by humans as a drug plant. It's used as a source of this high tensile strength fiber. And we actually think that maybe the first uses of cannabis were more related to the fact that this bast fiber, and bast fiber refers to the fact that these fibers are these sort of strengthened strands of fiber that are associated with the phloem. Phloem is like kind of the pipes that transport sugars in plants, and they're on the outside of the stem. So if anyone who's grown cannabis, if you've ever, even like a garage grow up or whatever, you've tried to rip a cannabis plant like this, rip the stem apart, it's almost impossible. You have to like saw it, cut it with knives, chainsaw it, whatever. It's because the bast fibers are like super strong. So cannabis is also this source of fiber, and it produces seeds that are really rich in amino acids and fatty acids. So it's quite a nutritious, hemp seeds in this case are quite nutritious. What more to say about cannabis?
We know like where it evolved and where it was first domesticated.
Yeah, it's generally believed to have originated in sort of Central Asia, I guess, northern India, Afghanistan, Iran, that kind of area, or Western China is another location that's talked about. And I think that's, a lot of plants where their highest diversity occurs, that's sort of the center of origin. And that's kind of where I think, both the historic distribution before humans started trading it and moving it around the world. So it originated in Central Asia. Its domestication is, it's still in the process of being domesticated, right? Like, as a crop, hemp is not really much of a domesticated crop. There's a lot of variability there. It doesn't have all the sort of standard crop type features that, in terms of like very stable lines that say corn or canola have. But humans, I think, there's evidence from China of a couple thousand years ago when it started, hemp or suggestion of hemp started showing up in sort of medicinal plant kind of descriptions. There's this famous case of and I've often shown this picture of a mummified, what people think might have been a shaman or high-ranking official. The grave was discovered in Western China in an area that's very arid and dry, and that preserved the grave. But this person, I think the date is 2,700 years ago, so quite early in terms of culture. But they were buried with a whole bunch of different grave goods, which included something like 750 grams of dried cannabis flowers. And there was a nice paper done. Ethan Russo is one of the authors, I think, where they analyzed some of the chemistry of this preserved plant material and showed that it had cannabinoid derivatives, I guess, degraded cannabinoids present. So the argument was that it wasn't just happenstance that this was a nice plant or the smell was sought after. It was probably used for drug purposes back in those days as well.
And so when cannabis, we use it for a variety of purposes. You mentioned there's the fiber, there's the seeds that can actually be eaten, and then there's the consumption for psychoactive and or medicinal effects. Can you talk a little bit about the part of the plant that is actually producing the THC and the other things? What part of the plant is that?
Right. So this was when cannabis makes cannabinoids in almost all parts of the plant. So you could, if you have very sensitive analytical equipment, you can detect low levels of cannabinoids, even in roots. But the most of these, the highest abundance of cannabinoids are actually found in the flowers, generally the female flowers. The male flowers are not as chemically interesting, but there are cannabinoids in male flowers as well. But it's the female flowers, and they have this coating of trichomes. So trichomes are epidermal hairs, so epidermis being the skin of the plant. So it's like an outgrowth of the skin of the plant, where and in general these are glandular trichomes. So female cannabis flowers will have a high density of stalked glandular trichomes. So there's a little kind of multicellular stalk, which is topped by what is essentially a little chemical factory. And I've probably overused that description over the years, but it's really true that what's happening is that those trichomes are not photosynthetic. They're not, using sunlight to make energy. They're actually just consuming resources from the rest of the plant. And they're at the top of the stalk is a little disc. For Canadians, I often called it a hockey puck. It's a little, almost like a little disc of cells that are completely committed to making cannabinoids and terpenes, which are the two sort of main classes of chemicals that are found in trichomes. And they're just, receiving sugars and nutrients from the rest of the plant. They're converting them into cannabinoids, and then they're exporting them, somehow secreting them out of the factory, which are these discs, into what is a storage site that's kind of like a bag or a balloon connected to the disc cells. And that's this sort of sticky resinous material, which is predominantly cannabinoids, but also terpenes, is then held there. And that balloon of metabolites is encompassed and protected by a cuticle, and then some cell wall components as well, which is still being sort of studied. So it's kind of like almost like a supermarket shopping bag. It's kind of this thin but strong layer that protects the metabolites and basically protects them from being washed away by the rain, being knocked off by the wind. But they're likely there because arthropods, insects, will come along, and their hard exoskeleton will break open those trichomes, and then all this goop, this runny resinous material, which is again probably pretty bad tasting and sticky, then just gums up the insect. Or I guess if you're a mammal and you're coming along and munching on that female flower of cannabis, you're also being exposed to those chemicals. And I think it's important, for listeners. The fact that trichomes are like the major site of cannabinoid production, but it's not exclusive. There probably is a small amount of cannabinoids found in leaves and other tissues. That's one thing. And then going back to what we talked about before with the morphine alkaloids, that if it's the female flower, that's where, if it's pollinated, at least seeds are going to develop. And I said before, the seeds of cannabis are rich in fatty acids and amino acids. They're really a little nutrition bomb. And so if you're an insect or giraffe or whatever, you want to get at food like that. And so the plant really has to protect those. And so the female flowers is like studied with these trichomes that are externally facing, right? They're facing the external environment. And they're really saying, like, don't eat me. If you come and chew through this female flower, or you land on the female flower and you want to get at those developing seeds, you're going to get a whole bunch of sticky terpenes. You're going to get these cannabinoids. Maybe the cannabinoids interact with your receptors or your nervous system.
Probably taste bad.
Certainly, yeah, they're going to taste pretty bad. And you're probably not going to eat your way through and get all those seeds. And so the plant, in many ways, is protecting its investment in its reproductive future.
And so when someone has a bag of weed, or they're looking at commercial cannabis flower, the tiny little crystals that you see, or the stickiness that you might comment on, that's the trichomes. That's where all of the chemistry is. all the chemistry that the consumer is really interested in.
Yeah. And so people talk about, how cannabis, like dried flower, say in a retail store or dispensary, it glistens, it's shiny, there's crystal. They're seeing trichomes. I mean, now I think consumers are getting much more educated because of just the way the cannabis world has changed. And so they know that word trichome, and they know they're looking at trichomes. But, really, if you see lots of trichomes, I think it's reasonable to expect there's lots of cannabinoids. The potency of the material is good, the quality is good. But yeah, you're seeing, when you go into a cannabis retail store, you're actually getting a little lesson in plant biology and botany by looking at those plants.
Interesting. And the flowers are not actually producing THC directly, right? So what are they making directly?
Yeah, so they're making the acidic forms of cannabinoids. So the biosynthetic pathway leading to the major cannabinoids essentially starts with basic building blocks of metabolism and constructs these elaborate molecules. But the way it does it is it creates an acid version. So the biosynthesis leads to tetrahydrocannabinolic acid, not tetrahydrocannabinol. So it makes the acid form of THC, often referred, you see it in the literature, THCA. And similarly, cannabidiolic acid is made, so the carboxylic acid form of CBD. And cannabigerolic acid, not CBG, not cannabigerol. And so the plant is actually full of acidic cannabinoids. And there is a small amount of neutral, so we would call the THCA, CBDA, CBGA, etc., the acid forms, and then the THC, CBD, CBG, the neutrals. And it's a non-enzymatic conversion. So it's not an enzyme that's making THC from THCA, or at least we don't think it is. But it occurs via just normal chemical processes like heating, etc. And so often when we analyze cannabinoids in fresh cannabis, you see almost all of the material as the acid forms, but there's a little bit of neutral material in there. So there's a tiny bit of THC always present.
Mm-hmm. So that's really why cannabis is traditionally smoked, or if it is eaten, it's actually baked first. It's to transfer the acid form of the molecule into the neutral form.
Right. So this is called decarboxylation. And, that's another term that's really entered the sort of popular lexicon because of the importance of that reaction. So yeah, the acidic cannabinoids are fairly labile, meaning they're quite readily converted into the neutrals, and that's occurring when you combust cannabis or you vaporize it at a high enough temperature, and that thermal energy is enough to kick off the carboxylic acid. It's lost just as CO2 and or carbon dioxide and leaving THC. Or you can do this, by baking or producing butter where there's heat involved and there's enough time and heat to make the neutral forms. And this also just goes back to that whole sort of ecology side we were talking about, where if what we know about the pharmacology of these two different types is that it's the neutral forms that are mainly active, or at least the psychoactivity side is that THC is the molecule that imparts it's the ingredient that's psychoactive in cannabis, or the primary psychoactive ingredient. It's not the acid form, and the acids don't bind the receptors, the cannabinoid receptors, and I'm sure we'll talk about that stuff as well. But so, the plant is making something in a form that's not the form that binds mammalian cannabinoid receptors. Mm-hmm. And it's not like, if you thought THC was made or cannabinoids were made as a defensive, for their defensive purposes, and it was because they interacted with cannabinoid receptors, say invertebrates like mammals. But then it means like the cow or the giraffe or the donkey or whatever, that would come along and have to heat up the cannabis in order to get it. It just doesn't make any sense.
Yeah.
So, and I think, the presence of cannabinoid receptors is not, I'm trying to remember if they even occur in the insect kingdom. I don't think they do.
I don't think they have CB1 receptors, no.
Right. So it's kind of like this whole, it's kind of crops up in people's comments once in a while. It's like, oh yeah, cannabis was trying to spread through the world by getting people stoned, and therefore 70 million years ago it evolved THC. Well, it didn't. It evolved THCA, and it's not even really binding the receptors without decarboxylation.
Yeah, I hear that a lot too. So the more plausible story from what you've told us is the plant, just like many plants, is trying to protect its reproductive potential, its seeds. It's making chemical compounds because it doesn't have the option of running away or something like an animal does. And these chemical compounds they happen to be psychoactive when they're converted, but it's really just trying to protect itself. But in the case of human consumption, we take the plant, we harvest the flowers, we process them, and then we do end up heating them to consume them in some way. Right. And we obviously have psychoactive effects with THC. So what is actually going on there? How does THC actually cause its psychoactive effects?
Yeah, so THC was discovered as the main psychoactive component of cannabis in the mid-60s in Israel by Raphael Mechoulam. And historically, it took a really long time to figure out how it exerted its effects, because what we know about many compounds, some of them poison, some of them with medical function, or in some cases both, was that alkaloids like morphine and cocaine and strychnine nicotine would all exert their effects by binding a receptor. But with THC, after 1964, it was like, well, how does it do all this stuff? We know it has the effects of getting people, causing euphoria in humans, appetite stimulation, change mood perception, all that sort of stuff. But the pharmacological mechanism was unknown, and it was always considered possible that it bound a receptor, so a protein in our nervous system that would then receive a chemical and convert that binding into some sort of change in the neuron. But for the longest time, including when I was just finishing my undergrad and starting my PhD, it was thought that cannabinoids like THC basically inserted themselves into lipid bilayers in neurons in the same way that it would change sort of membrane fluidity.
Like cholesterol or something?
Like cholesterol. I mean, ethanol seems to have a component of this as well. And so somehow that would lead to these changes in neuronal function that would translate into psychoactivity. It wasn't until I'm going to say it was that 93, Lisa Matsuda at NIH discovered the CB1 receptor. So the idea that was that people were looking for receptors or were cloning orphan receptors. So this age of molecular biology was hitting neuroscience at the time. And they cloned a receptor for which no known ligand, so no binding agent was known. And they started screening compounds that were in some chemical library, and they found a synthetic cannabinoid that was really able to tightly bind a receptor. And so that was the discovery of the CB1 receptor. And I'm going to say 92, 93. And so really, that's the history side. But what's happening is THC is entering, if you consume cannabis, THC enters your bloodstream, it goes into your nervous system, mainly your brain, where there's CB1 or cannabinoid 1 receptors in high abundance in the nervous system. I think it's actually one of the most, or not the most abundant receptor in our brains or in the brains of mammals. And it binds those, and this receptor then causes, sort of triggers a signal which dampens down neuronal function. And this dampening down of neuronal function just has an overall contribution to the kind of, I guess, the function of the nervous system, and causes the changes that we associate with THC intoxication or cannabis intoxication. Again, alterations in mood, perception of time, appetite stimulation, and things like that. So it's actually the plant molecule binding the receptor in our nervous system that's the important part of how it exerts its effects.
So the main effect of THC with respect to getting someone high and causing the classical effects of cannabis comes from its ability to bind and activate the CB1 receptor. What is CB1? What is binding to CB1 normally? What's going on inside the body when there's no THC around?
Right. So obviously, we don't have the most abundant receptor in our brains just in the hopes that you're going to consume some cannabis, right? It has other functions. And so back to the history, after the CB1 receptor was discovered, then the hunt was on for its endogenous ligand. So it's ligand, its binding agent that occurs in its normal function. And this was also work done or led in the lab of Raphael Mechoulam in Israel. And he had a scientist or a postdoc at the time, William Devane, who did work to actually, I think they worked on pig brains. So they were attempting to isolate the chemical from these brains that would then bind to and activate these receptors. And they ended up finding a lipid signaling molecule called anandamide. So it's an amide, sort of almost a prostaglandin type molecule that would then was shown to be one of the endogenous ligands. And then a second one was identified pretty soon thereafter called 2AG. And so 2AG and anandamide are the main endogenous or endocannabinoid endocannabinoids that we have in our nervous systems.
Mm-hmm. So I often hear people say when they're talking about anandamide is the endogenous cannabinoid. It's inside of us naturally. And that word translates to something like the bliss molecule, because anandamide makes you blissful and CB1 is associated with bliss. Is there anything to that? What's the story there?
Yeah, I mean, the name anandamide was coined by the Mechoulam lab, or maybe Raphael Mechoulam himself, because it was an amide by its chemical structure. And anand is a Sanskrit word which translates into bliss. So it was, I think the idea was that, the effects of anandamide would therefore be some of the effects of cannabis in terms of, producing a feeling of well-being, et cetera. But I'm not actually sure if we've even done enough, human studies on anandamide itself, or if it's able, to get, can you inject people with anandamide, or if that's ever done to sort of even determine if it's bliss producing. Mm-hmm. But I think that the circumstantial evidence is what lines up to call it the bliss molecule.
Interesting. So THC binds the CB1 receptor. That's what gets you high. That's why cannabis has its classical effects. There are compounds inside us naturally that also do that to regulate important aspects of our biology all day long. We know that some of the classical effects of cannabis are euphoria, appetite stimulation, other things. You can have side effects as well. You can have people get sleepy on cannabis. Anxiety and paranoia are often talked about as a side effect. What about habit-forming potential and addiction? Can you speak a little bit about what we know in terms of the habit-forming potential of THC and cannabis products generally compared to other common drugs, both licit and illicit?
Yeah, so this is not formally my area of expertise, so I'll kind of, give you the information that I know. Mm-hmm. I think in general, the sort of published literature would suggest that something like 9% of regular cannabis consumers, and by regular, I think this is like, daily, higher amounts of consumption, will develop a dependency to cannabis, meaning they don't want to stop consuming it despite the fact that there might be, problems in their life or, workplace issues or what have you.
Mm-hmm.
But from what I've been told, and, I've obviously attended lots of conferences on cannabis and cannabinoids and heard lots of lectures in this area. That's kind of on the same level as say caffeine addiction, which many Canadians and Americans definitely are addicted. And the fact is there is a small sort of physical withdrawal from stopping drinking caffeine. So about 9% of people who are regular consumers, that's significantly lower than alcohol, nicotine. Nicotine is obviously one of the more addicting substances that we use in our society, and things like heroin and cocaine and harder drugs, if we call them that, have more potential for addiction. Mm-hmm. So in general, cannabis is I'm going to say relatively benign, with the emphasis on relatively, because there are people who have developed what's called cannabis use disorder. So this is where their consumption is potentially harmful or impairing their ability to work, or giving them relationship problems, etc. Mm-hmm. But they don't want to stop because they enjoy their cannabis enough.
Mm-hmm. So there's some habit forming potential. It's not that there's nothing there. It's not completely benign, but it's probably not as habit forming or it's comparably habit forming to common things like caffeine, probably less so than alcohol and nicotine. Because I like to talk about that because I hear so much from both sides of that. There's some people that still to this day are demonizing cannabis in a way that's probably very excessive. And then there's also people that say it is benign and good for you at all times, basically.
Right. And I'm not sure for the latter group who are generally saying cannabis is completely benign. I mean, I'm going to say nothing is completely benign. Mm-hmm. Certainly, caffeine seems to be quite benign, and it's in every corner store or what have you. And you're in Seattle. It's kind of like the cult of caffeination in many ways. But there are people who have to lay off coffee because of heart arrhythmias, and I saw some evidence before about people who are slow metabolizers of caffeine versus others who metabolize it quickly. And slow metabolizing of caffeine coupled with some heart problems can put you in the emergency room too, right? So and sugar is another one, and you mentioned cholesterol before. I mean, we just have to be really careful about all our substances, and I think the same goes for cannabinoids and cannabis.
Mm-hmm. And we've mentioned a little bit some of these other compounds that are associated with cannabis beyond THC, and you talked about the fact that the trichomes, the parts of the plant that are producing THC, are actually chemical factories, so to speak, that are producing a bunch of stuff. So what are some of these other cannabinoids, and what are these other kinds of compounds like terpenes, and what do we know about them?
Yeah, so big question. So the cannabinoids, and I guess it might be worthwhile introducing the kind of term phytocannabinoid here, just for clarification. I don't use that term very much, and when I publish papers, I don't often say that. But when we talked about endocannabinoids, endogenous cannabinoids like 2-AG and anandamide, those are the ones that we produce in our nervous system, versus the plant-derived ones like THC and CBD being being plant. So the phytocannabinoids are a group of, I'm going to say, approximately 130 metabolites that have been identified in cannabis, with the main ones being THC and cannabidiol or CBD. So those are the two most abundant cannabinoids. And then the other big, I'm going to say the big 6, but they immediately become the big 12 because each of them has an acidic version, or at least almost all of them. So you have tetrahydrocannabinolic acid, THCA, and you have THC. You have cannabidiolic acid, CBDA, and CBD. You have cannabigerolic acid, CBGA, and CBG. Cannabichromenic acid and cannabichromene, so CBCA and CBC. And then we have tetrahydrocannabivarinic acid, THCVA, and tetrahydrocannabivarin, THCV. So that's another one that you might see at a couple of percent in some cannabis plants. And then there's similarly a CBD version of that, so CBDVA and CBDV. And then THC it can be oxidized to a compound called cannabinol or CBN. And actually THCA can also be oxidized to cannabinolic acid. So those are the kind of main ones. But then so those are the ones that, when I say main, if you were to buy cannabis, in the store that's a few blocks from my house here and put it into a chemistry lab that could do the analysis, you might expect to see most of those big 12, or at least tiny bits of them. And then beyond that, you're starting to get into these cannabinoids that have been identified by scientists who have been working to find new derivatives, but they're not present in high amounts in all known cannabis, or at least the cannabis that I'm familiar with. And so we're just getting into these dozens and dozens of new variants that have, there's a methyl group added onto another position that's different, and then there's like a ring closure, and then there's a kind of a rearrangement that's happened, and so that chemical diversity that we need to get to that 130-ish number is often all this kind of cloud of very, very small, I'm going to say relatively insignificant compounds.
I see.
So that's the cannabinoids. Did you want to?
No, I was going to ask about terpenes. Yeah, what are the terpenes?
So, and I said that cannabinoids are primarily present in cannabis, other than these things like there's a liverwort, which is a little sort of leafy kind of mossy thing that's found in Asia, that has a cannabinoid-like compound in it. And there's a sunflower in South Africa that has a cannabinoid-like compound in it. But let's call them unique to cannabis for all intents and purposes. The terpenes, on the other hand, are chemicals that are found in almost all plants. In fact, it's probably is all plants in the sense that they're a chemically and structurally different group than the cannabinoids. They're put together in a different way, though cannabinoids have a terpene component to them. So that we'll talk about that if we get back into biosynthesis. And so terpenes are often the smells and tastes that we associate with plants, like the pine smell of pine trees is a terpene, is alpha and beta pinene. And many plants that we use in perfumery, like lavender, those are terpenes as well. And so the cannabis plant makes terpenes as well as the cannabinoids, and it mixes them together, as far as we know, in those trichomes, those trichome heads, those little compartments, or big compartments in some ways, that contain the goop. The terpene components of cannabis are composed of two structural classes. So there's the monoterpenes, which have 10 carbons, and sesquiterpenes, which have 15 carbons. And that's starting to sound a little bit too much like third-year organic chemistry, probably. But it's important to know, because if you're a cannabis consumer and you go down to a shop and they're starting to talk about chemicals like myrcene. So myrcene is really the most abundant terpene found in cannabis. It's a monoterpene, but people talk about beta-caryophyllene, and that's a sesquiterpene. And so they're different chemicals. What is happening with the terpenes in cannabis too is that they're a highly variable and highly complex mixture. It's, when we look at the cannabinoid chemistry of normal cannabis, it's available for purchase in Canada or Washington State now. You're going to see a lot of THC, or as its acid form. You're going to see some CBG, you might see some THCV, a little blip of CBC, but there's kind of just a few. While the terpene composition of that cannabis is going to be 30 or 40 compounds that are readily identifiable. They're going to be, dominated by chemicals like myrcene, pinene, linalool, limonene, beta-caryophyllene as the major players, but a whole bunch of other ones, which is, I think, why cannabis, the smell, because that's a very, one thing I should have added before is that cannabinoids don't have a smell. Like when you smell cannabis, you're not smelling THC. It doesn't have an odor. It's not volatile. What you're smelling is the volatile terpenes that are being released by those trichomes that are maybe broken, maybe opened up a little bit. And those terpenes are then, moving into the airspace around the plant, and you're smelling those. And that's really what's responsible for the unique, terpenes are responsible for the unique odor of cannabis.
So you've got two classes of compounds. You've got the cannabinoids that includes THC, CBD, and these other ones. And then you've got the terpenes. These are the aromatic compounds that smell. The terpenes themselves have two major types. And what I'm starting to hear more about from people, both in the scientific community, but also on the consumer side in the cannabis industry, is this concept of the entourage effect, that the actual, the sum total, the effects, the full psychoactive effects you're going to get are not just due to THC. They're due to a combination of THC, potentially other cannabinoids, and potentially terpenes. Is there any truth to that? Where's the knowledge today that's scientifically defensible?
Yeah, I've often added to the entourage effect the word hypothesis. That this is a suggestion, it's a hypothesis that attempts to explain some of what we know about the effects of cannabis. And really it comes from the fact that people consuming different types of cannabis that would have different, even if there's THC as a dominant molecule, there's different minor cannabinoids or different amounts of, say, CBG in there. And then also different profiles, different composition of terpenes. That why, when you smoke a joint of one type of cannabis with a certain sort of chemical composition, would it maybe make you feel sleepy, while a joint of another type of cannabis that has a different chemical composition might keep you awake, right? So this has led to the idea that it's not just the major, not just the amount of THC, it's the other chemical components that are co-occurring with THC that are contributing to the pharmacology. And I'm going to say it's a very reasonable hypothesis. And I am generally a kind of a proponent of the possibility for this hypothesis to explain the subjective effects of cannabis, and why it's not just the sort of, let's call it the recreational, the kind of pleasure producing effects of cannabis. People who use cannabis for medical purposes also would say the same thing. Like there's a type of cannabis that really is great for their pain, and yet there's another type that is not as effective. But the sort of bulletproof scientific evidence so far is just not there. So we're accumulating information, and there's been some papers just published in the last year or so about the pharmacology of their minor cannabinoids. For example, cannabichromene, CBC, which is a little bit present in almost all the bud that people are buying in California and Canada and places. Is it contributing something that kind of modifies the effects, or is there something that terpenes do that is, one question would be, do they modify the ability of, say, THC to affect the cannabinoid receptor, the CB1 receptor?
Mm-hmm.
And there's recent papers that seem to say that is not the case, right?
Mm-hmm.
There's out of the Lambert Center in Sydney in Australia, but there still remains the possibility that terpenes, when inhaled, therefore, moving straight into your bloodstream,
Mm-hmm.
and reaching sort of higher concentrations, maybe they interact with other receptors, which then
Right.
have their own sort of additive effects onto the psychoactivity. So I think we're not there, and I use the word entourage effect with a lot of caution and a lot of caveats around it. But I think it's a plausible hypothesis to explain what I think many cannabis consumers have experienced, is that the different strains, different types of cannabis have different effects on them.
Yeah, and a lot of people say that. I do want to talk about these different types or different strains, the way that we think about.
Could I grab you just before that? And I just wanted to address one other thing, which I think is kind of maybe a little bit where we've been too focused on cannabinoids and terpenes. The fact that there's other components in cannabis that contribute to the smell. I mean, I think there's a little bit of it's still a kind of a bit of a black box, in the sense that, when you smell individual cannabinoids, individual terpenes, say you get a bottle of myrcene, bottle of pinene, linalool, caryophyllene, terpinolene, what have you. And I've had this because I've, had these labs where we study this stuff for years, and you smell them, you're like, yeah, that's myrcene. Smells kind of like a little bit like an oily kind of slightly diesely kind of thing. And linalool smells like sort of perfume, like Avon kind of stuff. And caryophyllene is kind of like woody, those things, but they don't smell like, for example, skunky smells, like the really skunkiness of cannabis, where, you walk around the city of Vancouver some nights and you smell something and it's like, is that a skunk, or is that someone growing weed, or is that someone smoking some weed? Like, they're close enough that sometimes it's hard for your nose to tell them apart. The skunkiness, I am not convinced the skunkiness smell of cannabis is due to terpenes. I think there are other components in there, likely in this case thiols, which are sulfur-containing aromatic compounds. We know that sulfur is, that's where that skunk smell comes from in the animal. Similarly with cheese, like, so you talk about UK cheese, and it's kind of like, literally it's like the Kraft Dinner flavor pack, right? It's got that tangy cheese smell. And that's not terpene based. That's the same kind of, whether they're aliphatic esters or alkanes or something that is present, that's the cheesy smell. Another component that's non-terpene that people are maybe aware of is the grassy smell. So if you have really, often it's associated with poor quality cannabis, which has been machine trimmed sometimes. There's this kind of smell that smells like a cut lawn or a hayfield just after it's been mowed down, right? And it's got this, it's a classic, it's a component that's produced by all plants when they're wounded. And really what's happening is that if you have a cannabis leaf and a manual trimmer, a hand trimming might cut the petiole, like the little stem that holds the leaf, while a machine trim might just cut it right, that little leaflet right in half. That damage site is essentially the same as when you mow your lawn. And that damage causes enzymes to cut fatty acids up, and it releases a C6 carbon volatile, which is, apparently we're really sensitive. Humans are really easy to able to smell these things. And those are other compounds that give cannabis its smell, which nobody talks about because they're not terpene. So I just want to make the point that I think the next steps in analyzing cannabis are getting beyond, like, there are people still isolating new cannabinoids, and all the power to them. That's important work. And people characterizing smaller amounts of minor terpenes. I think we're going to see the next step is all the non-terpene volatiles will be kind of the frontier to look at.
Interesting. So there's still really a lot to be discovered in the chemistry of those trichomes.
Yes. Oh, yeah. And just even chemistry alone, but biochemistry, there's like seemingly decades worth of PhD projects just in those little guys.
And so we danced around this idea of people saying out in the world that are consuming cannabis recreationally and for medical purposes, people saying that there are different types of cannabis, different strains with different types of effects. So the idea is, cannabis is a diverse plant. It's got morphological diversity. So there's plants that look different from other cannabis plants. And it's got chemical diversity, meaning two different plants can have different THC levels. They could have other minor cannabinoids. They could have different sets of terpenes. How much diversity is there chemically, and how much is that diversity captured, or how much does it correspond to what we see in the cannabis stores with all of the strain names and the indica and sativa dichotomy?
Yeah. I mean, maybe I could come out right off the bat and say the indica sativa dichotomy where you have, it's present in every legal market now too, which is kind of disappointing in some ways, but it's kind of like indica being plant-wise, these sort of short, stouter plants, wider leaves or leaflets, and often considered maybe a bit darker green in terms of the leaf color, versus sativa, which would be this taller, lankier, more open plant with narrow leaflets. And then so that's a sort of canonical view of these two types. And then the effects that are attributed to those two types would be indicas would be more sedative, sort of sleepier, couch lock, versus sativas, which would be sort of energizing, euphoric, uplifting, stimulating. And then hybrids would be a mix of the two. And that you'd have this, I guess a plant that would look sort of intermediate, and the effects would be balanced between the two. So that's really how the cannabis world likes to classify their plants right now. I'm not a believer in this classification system. So that I wanted to put that out there at the start. But to go back to your first part of your question, which would be, do the products available to consumers currently in the major marketplaces where cannabis is consumed reflect the cannabinoid diversity or the chemical diversity that's sort of possible in cannabis? And it's kind of hard to say, in the sense that it may be that there wasn't very much. There was lots of chemical diversity, but the main types were like a THC type, CBD type, and a mixture of THC and CBD. Those would be the main cannabinoids. And that before humans started like intensively breeding this plant, those were the sort of main types, and there was some sort of standard terpene types. And now we've actually increased chemical diversity in the available types because people have had gas chromatographs to do terpene analysis. So they're looking for like unusual terpenes, novelty seeking, where people who got really tired of 22% THC Kush with high myrcene suddenly wanted a new flavor. We have obviously seen a resurgence in CBD as a kind of a component that was completely neglected for quite some time because it was non-psychoactive, and everyone was chasing CBD as the sort of market driver, and CBD was present in cannabis in let's call it kind of pre-boom, California collecting seeds and sort of growing cannabis more, for personal use or for commercial purposes in the early 70s or late 60s. But, there would have been CBD in Afghanistan and Lebanon and places like that, or CBD producing plants. But it was kind of ignored for a long time until the medical potential of CBD was recognized. So we're seeing, I think many more options in the marketplace around CBD, and similarly, the more chemical diversity related to terpene profiles in the marketplace now, because people are looking for that kind of diversity.
Mm-hmm.
Does that sort of get to the question you were asking in a way?
Yeah, I think it gets at it. I think a follow-up question I have is, with the chemical diversity that's out there today among high THC cannabis. So if you go into a cannabis store, there's a bunch of flower. That's usually going to be the most popular category that people are consuming. Yep. And there's a bunch of different types of strains, allegedly. And you can certainly smell differences. You can see differences in the bud. But among the 90 to 95% of that flower that's high THC cannabis with low CBD, do you think it's possible, with the diversity that's there at the chemistry level, that there are strains that are more energizing on average or more relaxing on average? Or is that unlikely?
Yeah, I said before that I believe the entourage effect is a plausible hypothesis. Mm-hmm. I think that it's possible that there are strains in there that for most people can reproduce an experience that could be called more sedating or more sort of stimulating. The difficult part of this is also that we have this plant with this high amount of chemical diversity, and we still haven't nailed down, like, if something is sedative, what is the component? Like, what, mm-hmm. What is it? Is it higher myrcene, or is it, lower amounts of CBG or something? But also the fact that people are as highly variable as the plant itself. There's, mm-hmm. They metabolize cannabinoids differently. They may have slightly different, endocannabinoid signaling states. They also have this whole, it sounds like Timothy Leary, like set and setting, right? They're mm-hmm. sort of like what they're expecting. And maybe there's a contributing aspect to this as like the placebo effect. So you buy something that's indica and Leafly says, mm-hmm. This is the supreme couch lock that we've ever used. And then you go and you smoke it and you're like, wow, I'm really feeling tired now, right? Because you've sort of had that kind of suggestion as part of it.
Yeah.
But to answer your question, I do think within that kind of this sort of smorgasbord of cannabis that's now available, there could be things that are consistently have effects that are more like sedation or stimulation.
Mm-hmm. And I've always wondered, how much of this stuff can be explained by dose-dependent effects from THC, because I would be confident in saying that we know as a fact, more or less, that cannabis has dose-dependent effects. And if you consume something at a relatively low dose, you take a puff or two versus you smoke a lot more, same exact product, you're going to feel very different if you take a lot of inhalations of something with THC versus just a little bit.
Right. Yeah. And I think, that the other side of that same coin is the effects of CBD. So I think there are people who sort of ascribe certain effects to CBD because how the genetics works is that you can have THC dominant plants or you can mm-hmm. have CBD dominant plants. And when you cross the two, you have, and they basically are because you have two alleles of the same THC producing enzyme or two alleles of the same CBD producing enzyme, or you cross them, you get one of each. So you have approximately equal amounts of THC and CBD. And so people consuming that would say, oh, I really found that CBD had this effect that I was way more clear. Mm-hmm. It's like, this gives me this mental clarity or something. Mm-hmm. And really what they're talking about is the total amount of cannabinoid in that product is kind of fixed. And so when you divide it in two, you're going to get, instead of getting 24% THC, you're going to get 12% mm-hmm. THC and 12% CBD. So the low dose of THC there had an impact on the effect of the consumption. And they said, oh, that's the CBD, when in reality, it was just a smaller amount of, or a lower amount of THC was the effect.
Hmm. How, so you mentioned a little bit about the genetics there. In one sense, the cannabis genome is a lot like our genome. We get two copies, one of every gene, one from mom and one from dad.
Right.
And I know that you used to be involved in a big project that looked at the first cannabis genome sequencing. So, can you talk a little bit about cannabis genetics and what we know there? How does that genome look similar or different to something like our genome or another crop like corn or something?
Yeah. So as you were said, there's sort of two copies. It's a diploid genome, and that's not always the case in plants. You can get, hexaploids, 6N, and so it's a relatively simple genome of about 825 megabase pairs, which places it sort of moderate size. Like
Mm-hmm.
the human genome is over 2 gig, right? So cannabis genome is smaller, but it's bigger than, say, Arabidopsis, which is the kind of lab rat plant of the plant genetics world, by a significant amount. The cannabis genome has proven quite, I think, difficult, partly because of the dioecious nature of cannabis, in the sense that generally you want to inbreed plants to kind of have a homozygous, meaning that the two copies of the genome are as similar to each other as possible, and that gives you an easier time.
Is that what they mean when you hear growers say stable genetics, or they're going to stabilize the genetics?
Yeah, I mean, they say that, but they're not. The growers are not really stabilizing the genetics except by clonally producing the plant. So the idea is that inbred lines, as we find in modern corn breeding and other things, will then, once you've crossed two inbred lines, will produce a more stable hybrid, F1 hybrid. And so that would be the goal of lots of breeding and seed production in other crops, but we're not really there yet in cannabis. So back to the genome, though, it's highly heterozygous, meaning there's lots of differences between the two copies, which makes sequencing quite difficult. And the other problem with the cannabis genome is it's full of repetitive elements. So there's lots of, let's say, sort of almost like junk DNA, just that's randomly repeated. And this is, old viruses and things that have kind of hopped in and out of the genome over the millennia, the millions of years that plant has existed. So it proved to be quite a challenge. It's still quite a challenging genome. And there's a couple of other factors. I mentioned the heterozygosity, the fact that it's still not a mainstream plant for people to work on, right? So, the US is only now coming to grips with allowing more research growing to occur in universities. So, the kind of mass weight of science focus that you put on either a crop species like tomato or corn or canola because of its economic significance, or conversely, the scientific focus that you put on a plant like Arabidopsis, because, as I said, it's kind of the lab rat, the fruit fly of the plant world. So there's just every lab working in plant genetics in the world is working on Arabidopsis, and so sequencing is focused on that plant. Cannabis is neither, even though we're talking about it. We obviously are, think it's a great plant, and there's, it's in the news more than corn or canola, definitely, but it still doesn't have a lot of people working on it. And that's just as an aside, that's one thing as a scientist that I've really sought to sort of encourage, is the fact that we need this kind of legitimate research ecosystem around cannabis that kind of treats it like a normal plant, that it's not necessarily trying to squeeze value out of it because there's some company interested, or studying it because it's the latest fad in the news or something. But the fact that it has these really unusual characteristics, it makes these amazing diversity of chemicals. It has these bast fibers. It's hemp seeds. It has this long association with human history. So there's this kind of evolutionary and cultural aspect to understanding, how we domesticated it. For all those reasons, I think cannabis should be brought into many more labs and have more resources around it. But I sort of digress on that. On the genome, though, I think it's kind of interesting about how this genome, the story of the cannabis genome, how it kind of came to be. Do we have time to chat about that? Probably, right?
If you're not time limited, I've got plenty of time.
No, I'm okay. My kids might come home from school and I'll have to kick them out of the room. But other than that, so I was talking about my research career in cannabis before, and how I got to Germany. I started working on this plant, and really those projects were very much focused on biochemistry, and I was trying to identify the enzymes of cannabinoid biosynthesis. So how you go from basic metabolic building blocks down to THCA or CBDA. And when I moved back to Canada, I had all intention of working on that project. So I took this job with the National Research Council in 2003, and I actually applied for the job with a job talk. As when you apply for science jobs, you're invited to come in and give a seminar. Gave a seminar on cannabinoid biosynthesis, and I was like, okay, this is going to be great. In 2003, Canada had just started to allow patients to access medical cannabis, so I thought it was a very timely kind of research program to start. And I went to the institute director in Saskatoon because he needed to kind of give me permission to apply for a research license in order to grow cannabis in the lab. And I went to his office and I said, hey, you need to sign a letter. And he said, I'm not signing a letter. We're not going to work on cannabis. We're the government. And I was like, because it was a federal government institute. And I was just like very deflated, obviously. And it felt like a major setback, but also just that I had been I'd almost been lied to, in the sense that I had been sort of recruited to this position with this idea that I'd be able to work on cannabis, and then they were denying that. So I ended up working on humulus, on hops, as kind of a substitute for a few years, and made some really interesting discoveries and things. But about 5 or 6 years later, I still was trying to figure out how I'd work on cannabis. And I got some research funding as an adjunct professor at the university there, and I started to sort of piece together a research program on cannabinoid production and biosynthesis. And that led to me, isolating trichomes and looking at what the genes were that were expressed in those trichomes. As I said before, those little disc of cells that are in the top of the stalk, those are what are making cannabinoids. And so the idea is that if you sequence all the genes that are expressed in those, if you could isolate those cells and sequence the genes that were turned on in those cells, you would have kind of an insight into all of the most important genes for cannabinoid production. And then we could use that information to start doing basic biochemistry and, expressing those genes in bacteria and trying to figure out what chemistry they could catalyze. So I did that. That was a very successful research program for a few years, and I'd done a bunch of work and published some work. And I was actually down in California in 2008, 2009, trying to crystallize one of the enzymes that I had from this research program, a enzyme that we thought was involved in making cannabinoids. And I was at the Salk Institute, which is like this, world famous biomedical institute in La Jolla in California. And I got an email forwarded to me from another researcher in Saskatoon it was actually from a professor at the University of Toronto, whose name is Tim Hughes. And Tim said, hey, I want to sequence the cannabis genome. Does anyone want to help? And I was like, well, of course I want to help. This is what we've always been dreaming of. And even years before, I had applied for money and to sequence cannabis genome and hadn't got the funding. So I immediately joined forces with Tim, and we started planning this project. And the idea also was that both cannabis was suddenly becoming really, really important economically, legally, in regulations, etc.
What year is this again?
2008.
2008.
Yeah. And then, and by that time, I have to say I was growing hemp in my lab in Saskatoon, but I wasn't growing drug type. Health Canada had permitted me to grow hemp, but they were not permitting me to grow drug type cannabis. So I got in touch with the one company in Canada at the time that was licensed to grow medical cannabis. And I said, hey, I've got this great project with the University of Toronto. We're going to sequence the genome of cannabis. You're being paid by the government to grow a supply of medical cannabis for patients. We should sequence the strain or the cultivar that you guys are growing because it seems to be the one. And they said, no, sorry, can't do that. That would infuriate Health Canada. We don't want to get in trouble. You can go somewhere else, basically. So I said to Tim, hey, we can't get any drug type cannabis, but we could sequence hemp from my lab. And he was like, come on, that's a joke. If we're going to sequence cannabis, excuse me, we're going to sequence cannabis, it has to be drug type. And I was like, yeah, you're right. So then I started to think about, how are we going to do this? And I came up with this kind of scheme, which was, I knew people in Vancouver who were growing medical cannabis as authorized patients. So the government had authorized them to grow cannabis. And I got in touch with one of them and I said, hey, would you guys be willing to donate some material into the sequencing effort? And they said, for sure, come and do it. And, what I sort of planned on doing was getting some equipment from the university in Vancouver, transporting it to their basement where they were growing the plants, isolating the DNA there. And the DNA was free of drug value, right? I mean, it's just DNA. Mm-hmm. So there was no control over it. And then transporting that to my lab in Saskatoon and using that. It didn't really work that way because I ended up having to get leaves from them. I couldn't get the equipment organized. So I just grabbed a few handfuls of young leaves of purple kush, which was the plant they were growing. And I put it in a cooler on ice. And I actually drove from Vancouver to Saskatoon, which is, in hindsight, probably not the best idea for like a federal government scientist driving across Canada. But, it's 18 hours to go driving from Vancouver to Saskatoon. But anyways, I did that. We brought the material into my lab. We prepped the DNA. The DNA was sent to the sequencing labs. And that was really, that purple kush sample was the source of the first cannabis genome that we published in, I guess it was November 2011.
2011. Yeah, I always did wonder why it was purple kush. And now I know.
Yeah. I mean, it seemed purple kush was widely consumed at the time. I mean, it's still around. It's about 16, 17% THC generally. And it was just kind of like, it wasn't exactly a lab rat kind of version, but it was pretty standard cultivar that was available.
How stable? So you mentioned it's usually 16 or 17% THC. How stable are the chemical profiles between different strains? So if someone was growing purple kush up there in BC and it came out at 16, 17% THC, they grew it again the next year. Is it going to be about the same level? If they gave a seed to someone else in a different province, how different would it be?
Yeah, there's two things there. One is you mentioned seed. So the seed from a plant like that is not going to replicate the plant from which it was produced. So it's a little bit like, people always said about apple trees. Like if you ate an apple and you put the seed and grew it, and you're eating Golden Delicious and you took the seed, it's not going to be Golden Delicious that grows from there. And the same thing, the problem with cannabis is that there's still a lot of diversity in those genomes. Like the stable genetics, say for purple kush, don't exist. And this is really why clonal propagation, so cuttings from mother plants, is the name of the game with commercial cannabis production, because you're essentially cloning. You're making genetically identical copies. So, if you took a seed of that purple kush, it wouldn't replicate the chemistry exactly. It might be close, and some plants are a little more inbred and more stabilized than others. But, the issue is this, is that cannabis, the genetics determine the kind of basic chemistry of the plant. So if you have a THC dominant plant like Purple Kush, it's not like if you grow it somewhere else, it's going to be CBD dominant, or it's going to be a mix of 1-to-1 of THC and CBD. It's going to keep that THC dominance. And the plant will be, physically, when you grow it in different places, it depends a lot on the environment. It can be short and spindly, getting a bit taller depending on lighting, etc. The terpene chemistry is probably going to be relatively set in stone as well. So say that Purple Kush, and I'm trying to remember the composition, but definitely it's going to be myrcene pinene dominant. So the two main terpenes are going to be myrcene and pinene. If you grew it somewhere else, it's still going to be myrcene and pinene dominant. It's not suddenly going to be linalool and limonene dominant.
Yeah.
Right. So the genetics determine that, but the amount of cannabinoids and terpenes produced is going to depend a lot on the environment. And we have certainly seen this in research programs that I've been running recently, where we've grown, plants in indoor settings under fairly controlled conditions, and yet the chemistry varies. You might see an 18% run, and then there's a 15.5% run, and then you move it to the greenhouse, and it's, in the summer it's 21% THC, and in the winter it's 17.5% THC, and this is one of the difficult aspects of modern cannabis production, is that even though the genetics kind of say this plant should be 20% every time, it depends so much. And by 20%, I mean 20% THC, and say it's 1.5% terpenes, and those terpenes are myrcene pinene dominant. But if it's not grown right, if the harvest is too early, if the lighting is inappropriate, they can change the chemistry downward quite a bit.
Okay. So it sounds like a lot of the profile, meaning the basic chemical profile, whether it's THC dominant or CBD dominant, whether it is a high myrcene pinene terpene profile versus a high terpinolene terpene profile, say, that's mostly determined by genetics. And the environment is really critical for determining the overall levels of cannabinoids and whether you've got as high a percentage as that plant is capable of potentially making.
Right. Yeah, I think the environment, and let's also add the sort of developmental aspect, because obviously you can have a Purple Kush, cutting this tall, it's not flowering at all, and its chemistry is going to be so different than 8 weeks away when it's fully flowered, or 10 weeks away. So there's that developmental aspect as well.
I see. And so you had this background in science that we've talked about. You had this background in genomics and plant botany and biochemistry. Can you talk a little bit about the time? What was your decision-making process, and when was it for jumping into starting a company? And how did you go from science to the world of startups, and not just going into them, but actually starting the company yourself?
Right. Yeah, no, it was, that's another whole sort of chapter. And that's the sense that a few things were happening for me in my, let's call it professional, but also my personal life, that kind of led to that decision. Though I'll add that early on in my cannabis science career, I was always thinking there was a potential company, like really a biotech company focused on cannabis. I just always had this belief that, it was not just a fascinating plant, but a highly valuable plant that deserved more study, but also that study could lead to economic gain. And there was some company embedded in there. And my mind turned to that idea at a couple of points in my career, even before I left Germany, actually. So I was kind of like, I had sort of thought about that. But after I mentioned, I started this job with the National Research Council in Saskatoon in 2003. And that had started out, it was a good job, but I had that early trouble trying to get permission to grow cannabis and sort of switched over to hops as an interim measure. But I had finally kind of made a bunch of gains research-wise, published the genome in 2011. The olivotolic acid cyclase came out around that time as well. This was a kind of a high point in terms of the biochemistry research program. So I thought I was doing well, but NRC at the time started to really change their focus. Where we had a prime minister in Canada at the time, Stephen Harper, who was not well, I won't mince words. He didn't like science. Like, he was, let's call him an anti-science prime minister. And he was, he tried to suppress information around climate change, and he had federal scientists remove the word habitat from their lexicon because it was helped endangered species. And he really changed the National Research Council's focus, where he said, you guys have to stop playing around with sort of curiosity-driven research. We need something that drives the economy. And I said, yeah, but cannabis is doing that. We've just got this, medical cannabis program for patients that's authorized by the federal government. There's a company growing medical cannabis. We don't know much about this plant. My research program is contributing to that. And NRC was kind of like, yeah, not really. If you want to do that work, you have to find some external money. So I really sort of felt under pressure despite those successes. But then the same prime minister came along in 2012, and he did something that was actually kind of positive for me, in that he and his government were unhappy with the way the medical cannabis program had gone for patients, because it had turned from a small number of patients to almost a full-scale industry of sort of homegrown cannabis being distributed to dispensaries. And the police and the government were not keen on that. So he said, okay, what we're going to do is we're going to take the ability of patients away, but we're going to allow companies to grow medical cannabis to supply those patients. And thankfully, the patients didn't lose that right, because there were some court challenges that helped that right away. But regulations came out in December 2012, which said, this is how we're going to allow private sector companies to grow cannabis for medical patients. And I immediately said, there's an opportunity here, in the sense that the regulations both would allow companies to grow cannabis for research, but there's testing required, there's knowledge about the plant. And the other part is, as much as I like Saskatoon, me and my family are from the West Coast, so it was kind of cold, dry winters in Saskatoon, and long, snowy winters that after a while we were sort of hankering to get back to the rain of British Columbia. So I decided at that point that, the research conditions were deteriorating for my program, but there was suddenly this opportunity to start a company. And I was like, so this would have been December 2012, those regulations were published, and the new regulations were going to start, I think, April 1st, 2014. So there was a bit of time. 2013 was a time of transition where my family and I left Saskatoon. We moved back to Vancouver. I got an adjunct professor position at the University of British Columbia at UBC, which allowed me to continue some of my academic funded projects. And at the sort of early age of 44 years old, I started a company that, with no business experience. Like, I jokingly say that I'd never even run a lemonade stand as a kid. Generally what I did for summer jobs was always sort of like digging ditches and planting trees and things. And I didn't really have a business background at all. And yet I plunged head on to a startup. And I had, in my discussions in Vancouver, I'd found another scientist, John Coleman, who's an organic chemist. also with a PhD from UBC, and he was interested in leaving his position and joining forces. And Anandia was born from just a lot of plugging away on a laptop. In effect, exactly where I'm sitting now, because I have the same dining table now that I did in 2012 or 2013. And so I basically just climbed to the top of the high diving board and jumped off and saw what happened. And it was just phenomenally perfect timing. There was two things that helped Anandia succeed. One is that we were one of the first companies to offer quality control testing. So the government was requiring product to be tested for cannabinoid levels, but also, bacteria and mold and heavy metals and aflatoxins. And so John Coleman, being a chemist, really had the expertise to set up this analytical program. And at the same time, we were saying, we have, this sort of biotech plant breeding side is really exciting. And this is the future potential. And Anandia was doing both. So that was great. And, nobody really had set up a cannabis specific science company before, at least in Canada. And many of the testing labs that were coming on stream were environmental or forensic testing companies that were adding cannabis to their repertoire. Mm-hmm. We were cannabis focused. And I was in this unique position that I had this kind of pedigree or research reputation. I published the cannabis genome a few years before. I had, cloned and published on many of the enzymes or some of the enzymes of cannabinoid biosynthesis. And there are still not that many people who had research credentials in cannabis science. And I was at the head of this company. So that opened a lot of doors and really excited investors and others who sort of joined in the Anandia journey.
So you had absolutely no business background, not even a lemonade stand. You were a botanist with a background in plant biochemistry. And in this case, I think similar to what you were saying earlier about how you got into cannabis research within your research career, you were really, you were adapting to situations where, one thing was winding down or not going the way you thought, and you had to find something new. And then you just sort of jumped at something that was adjacent that you noticed.
Right. Yeah, I think that's exactly it, that I was open to these opportunities that were not exactly what I had been planning, but were close to it. I will say that it was, moving from the research project that wasn't working to cannabis in 1998 or early 99, whenever that was in Germany, was a kind of more standard thing. By the time I was switching from the federal government to starting in Anandia, I had 2 kids. We owned a house in Saskatoon, 2 young kids. They would have been, grade 3 in kindergarten or something. My colleagues at the NRC, when I told them I was leaving, they looked at me like I was an alien. Like the fact that you would leave a well-paid government job with essentially what people call the golden handcuffs being this great pension that will come to you at the end of your research career or your career with the federal government. And I was giving up all that and going to a city, which is one of the most expensive in North America for housing, at least, to start a company. And with they probably thought I was a good scientist, but they certainly didn't think that I could run a business. And so I did. I saw that opportunity, and I don't really. Startups is a strange thing in the sense that a startup world is a culture unto itself, right? Mm-hmm. And I go to UBC, and there's an entrepreneurship at UBC Center, which I actually spent some time using their facilities, and they were very, very welcoming and helpful. But there'd be these young engineering grads or software guys or maker lab people, and they take their cues from Silicon Valley and that kind of the idea of what a startup is. And I essentially, not that Anandia wasn't a startup, but I essentially ignored that. Like I didn't really sort of follow this sort of standard startup sort of approaches and guidelines. I think I had my own idea of what I wanted to do at that point.
Mm-hmm.
And I really, John Coleman was an excellent business partner because he complemented a lot of the stuff I did. And we got ourselves like a great lawyer because, a lot of what you do in business is deal with legal agreements, and whether they're employment agreements or non-disclosure agreements or what have you. And this lawyer was referred to us right at the start, and he incorporated Anandia like November 26th, 2013. And he was a lawyer when we sold the company to Anandia in 2018. Like he did all that. And we recruited sort of wise men and women to advise us. And I always, whenever I didn't know something, I would just seek out expertise and opinion and advice. And I'm not going to say it was easy. It was actually extremely stressful. Like, there were times when in India, we had sort of run down the bank account, and we'd be meeting with an investor / advisor here at a restaurant in Vancouver for breakfast. And he'd say like, guys, you've got like 2 months worth of money left. And, at your current cash burn, what are you going to do? And we leave that breakfast, and just John Coleman and I would look at each other, and we'd both be like pale and green, because you knew, you were going to have to lay people off, sell equipment, maybe the whole thing was going to go belly up. So, startup, it worked because of the timing and partly because of my previous expertise, but it wasn't a sure thing.
Beyond the actual science, beyond having the scientific expertise to run the scientific instruments and things like that, do you feel like your training as a scientist and as a researcher prepared you in any way for being able to manage a startup?
Sorry, I don't have COVID. Yeah, I thought about that quite a bit. And I think there's many, I don't know what we call this, sort of not biases, but assumptions about scientists that you wouldn't have the skills to be in business. And I think there's a bunch of things that I learned to do. I'm not going to say I had some talents probably to start with, but I learned over my science career. Certainly building teams is like what all scientists do, right? You're building research groups. Outside of your research group, you're building collaborations to get things done. So there's a lot of both relationship building, developing the expertise of people who work with you, mentoring those people, managing human resources, which as CEO of Anandia and then subsequently when I worked at Aurora was always like, I'm going to say 30 to 35% of my time. You're just managing people. I did that before too. At wherever I was working, you were always, talking grad students off the ledge when experiments weren't working, or cajoling colleagues to join forces in a grant or something. You're just, so people skills are everywhere. And so scientists have lots of the same skills.
So when you say managing people, you mean just having those one-on-one or one-on-two conversations with individuals to help get them through whatever they're working on that week, that type of thing?
Yeah, I think it runs the gamut between working, meeting one-on-one with people who have both, technical issues or maybe just motivation problems or questions about where things are going, to, meeting in whole groups that, sort of cheerlead the team and bring everyone up to speed on a new direction or introduce new team members. Like, I think there's always this mix of like the sort of individual contact, because if you only talk to say you had 10 people working in your lab, whether it's a company or a university environment, and you're just talking to them one-on-one, you need that sort of shared communication in the group setting as well, and maybe even smaller groups. So, but I guess when I'm managing people, it really is more what you were suggesting, which is like working with people on an individual level to get the most out of them, but also, stressed employees or upset employees are never going to be very productive. So you're trying to figure out how you can help those situations, maybe even prevent things from happening and boiling over before things get too bad.
One of the things that I think translated well for me from going from academia to the private sector was actually public or just group presentations of complicated things. So in some ways, when you're doing academic research, you're sort of always working towards telling some group of people what you did. And I wondered if you could comment on that and talk a little bit about your funding at Anandia. What was it like raising money? And do you feel like you were prepared for that in any way from your scientific background? Was that extremely awkward, or did it come fairly naturally to you?
Yeah. I mean, this is going to sound, like I'm a sales guy at heart or something, but I found that when I was an academic or presenting research results at, say, conferences or seminars, that I was kind of, you're selling something. You're telling a story, and that story, of course, is based on data and evidence and polyacrylamide gels or HPLC traces or something. But there's a story there, and so I think I learned how to present scientific information in a way that was both, at a level that people could digest and interpret, but also it was memorable and people could sort of follow the story. And I think those sort of skills helped in fundraising. Certainly, that's what I did a lot of, is pitching to investors. It would be online, it would be phone, it would be in person, and giving lots of talks and panels at investment conferences and cannabis meetings about what Anandia was doing and what we planned to do. And I took to that very naturally. Like at one point, John Coleman and I, so this would have been, I'm going to say this would have been December 2017, maybe. We went to Toronto. So Bay Street is the financial hub of Canada. It's the main financial center in Toronto. And we went to Toronto to raise money. And it was like sort of December 18th or 19th or whatever. And we flew out of Vancouver. And when you leave Vancouver in the morning, you get to Eastern time zone. It's like 4-hour flight or whatever. So you're getting there early evening. We had some dinner, and actually we got there later than that. And we got to this hotel downtown and the power was out. And then in the morning, in the middle of the night, after trying to get some fitful sleep, the fire alarm went off and woke everyone up. And then the next day we got up and we were meeting investors to raise money for Anandia. We had like a 8 a.m. breakfast and a 9, a 10, 11, a 12, a 1, a 2, a 3, a 4, a 5. So there'd be like, 11 meetings or something. And I said the same thing to each meeting because they'd ask the same questions. I would, give the same answer. We were trying to raise money for the expansion of Anandia. And we got out of there with very little commitment. And it wasn't a bust, but it was kind of like, where's this going? By like January 4th, or just 2 weeks later, I think we raised $13.5 million from that one trip. And so it had been extremely successful. And that was money that was going to fund the growth of Anandia. We're, taking on new lab space and hiring new people. And I mean, I think that's testament to the fact that after a while, I really learned this sort of investor relationship business fairly well. And I think that stemmed from the fact that I had spent years selling myself and my ideas in a scientific context.
I love this story for so many reasons, but one of them is I see so many, like I have a science background. So obviously I love that there's this, what I'll just call a science company or biotech company that was so successful and was doing like interesting work from a biology perspective. But I've seen so many people come into the cannabis industry from other areas, what I'll just call business people. So investment banking types or Silicon Valley tech people. And they have like their own language and their own set of goals and their own styles. And they often say things like what kind of exit they're going to get. They talk about the exit and how to structure the exit. And that just means, selling the company or going public. It's sort of really, it's almost like a coded way of saying, how can I make an incredible amount of money as quickly as possible? And yet you just told us a story of this botany guy who had never even run a lemonade stand, and you end up starting a company and selling it for something like $100 million to a larger cannabis company. I'm assuming, based on what I've heard so far, that wasn't necessarily your intention. You didn't start the company and have like a specific strategy for how can we get that type of exit. So what was the story on how the sale of Anandia started to come into fruition?
Yeah. I'll say a couple of things about an exit before I say how Anandia sale came together. Many investors, when they're investing in your company, they want to know what the exit is. If they're going to give you, I don't know, $500,000 and you say, well, we're just going to grow the company and see what happens, or we're going to grow the company and we're going to dividend out money once we start making something, making some real revenue, they're going to be like, well, that's not what they were looking for. Mm-hmm. And I was always quite, I'm going to say, coy about this. Like I didn't found Anandia to sell it. And it's not like I didn't have that in mind. And, when we started Anandia, I mentioned a lawyer who we worked with start to finish. We put together all the right documents. We had a very clean sort of file on Anandia around ownership structure and the board structure and approvals and things like that. So we kept the clean house, which was, I think, really important because, when your company grows, if you didn't address those issues early on, they just become worse and worse and more difficult. But I built Anandia because I really believed in the potential of cannabis. I believed in the sort of fun and the curiosity that I had, and I gotta do something on my computer there. And I mean, by fun, I mean not just like smile on my face. This kind of passionate sort of focus on doing what I was doing. And I probably didn't want to talk about an exit because I didn't want it to end, despite the fact that I said it was really stressful at some times too. And if I was an investor and I saw that in a founder pitching me, I might, there might be some investors who say, oh, that guy's a waste of time or that company's a waste of time. But there's others who would say, that's the kind of passion we need to make a company succeed. And so I think that kind of my sort of not addressing exits and things like that, not that was by design, but it was just, that was what I was myself thinking, was sort of disarming. And they sort of said, well, that guy's fully committed. That's great, because I want my money will be in good hands to be fully committed on what they're doing. So the sale of Anandia came about to Aurora Cannabis over quite a few steps, in the sense that Aurora always had and still has a really strong focus on science, and sometimes science backed by as it leads to regulatory compliance. But also their quality assurance and quality control was very, very driven by high quality science. And so Anandia started testing for Aurora early on, as we tested for a number of producers. So we had early contact with Aurora through that testing relationship. So they got to know us through that, and that led to lots and lots of closer discussions with their executives. The CEO at the time was Terry Booth, who I got to know really well. And there was just, a lot of, I think, good chemistry between me and John Coleman and the other leads at Aurora. So we kind of had this, intentionally arm's length relationship with Aurora, because as a testing lab, you're working with their competitors as well, and you don't want to sort of cozy up too much with just one client. But we got to know them, and we actually worked with them through some tricky business as it related to not they having pesticide contaminated material, but them looking at another supply of cannabis that turned out to be contaminated with pesticides, which actually, it's kind of an untold story of Anandia, but our work on pesticide analysis in cannabis really led to like a front page news stories around contamination of some producers. Again, Aurora was very safe and very clean, so there was no question there, but others that had to have their product recalled. Health Canada, who's the regulator in Canada, had to change their regulations rapidly in order to prevent further contamination by unapproved pesticides. And we essentially had run some samples on our mass spec in Vancouver that had detected these pesticides. So, Anandia was really leading the way in a lot of very important things around cannabis, and Aurora was recognizing that leadership. And so, as the sort of cannabis boom accelerated, I think Aurora was like, what are the assets that can really help us outcompete other large players, but also have this sort of global footprint that we're seeking. And Anandia, they were, it's public record, they bought numerous producers at the time. And Anandia was a relatively modest purchase at around 100 million. I mean, it seems like a lot of money now, but Aurora's worth billions, and the cannabis industry was just skyrocketing from there. And bringing me and Anandia and John Coleman into the Aurora world made a lot of sense. I mean, I was happy because I just left Aurora. I left my position as CSO, Chief Science Officer, not quite a month ago, but I'm still a strong supporter of this company. I love that. I love what Aurora's doing, and I recognize they and many other companies in Canada have made some mistakes along the way as we've adjusted to this kind of very fluid marketplace. But I was really happy to sell Anandia to Aurora. I say that at the same time, I mean, there's always this kind of seller's remorse. I think this is where, if you talk to any other startup founders, I mean, you've essentially been the boss, right? I mean, I was the CEO. John Coleman was a chief operating officer of Anandia, and we had a board, but the board was like investors, and they were kind of happy to support most of what I was planning as a leader. And so I was, I kind of love autonomy and independence as a person, and I've often, that's great. And when you sell your company, you're no longer autonomous or independent. You've got bosses, and those bosses have a board as their boss, and those boards of a public company have real teeth, right? I mean, they're there to ensure that shareholders are protected and the company's reputation is protected. And so, you don't mess around. And so suddenly you're in a whole other world. So I kind of think of my career. I mean, Anandia was like one section. Joining Aurora was another beast entirely. And I probably learned as much or more working for Aurora after Anandia about how companies work and how corporations work, and how big companies are organized as well.
Interesting. And so all of this, there's like your personal story that turns into the Anandia story and the Aurora story, but then all of that is happening obviously in the context of Canada legalizing cannabis. And so we saw this whole industry start. We've seen rules and regulations evolve, and so on and so forth. Let's talk a little bit about Canadian legalization. What so far has surprised you about how things have gone for Canada in this first early phase of legalization? Did anything go as you expect, or maybe not as you expect in a big way?
Yeah. I mean, a few things, observations. So there was obviously lots of concern. The sky is going to fall. Our roads are going to be, inundated by stone drivers, and accident rates and car accidents would go up. Um, that kids would have more access to cannabis, and youth rates might go up. These would be like, sort of war on drug crusader kind of perspectives. But, in some ways, it was an experiment, even though, Colorado, Washington State, other places had legalized, not at the federal level, and not a whole country in one fell swoop. Um, and nothing like that has happened. The police, I think, it's been a little while since they've even commented, but they haven't seen a huge increase in stone drivers and road problems. I think youth rates might even be down a little bit. I'd have to check the stats, but I mean, it's negligible. So, I mean, that's been, I guess one thing is that there's been some more cases of, let's call it, inadvertent cannabis consumption, like children or adults eating a gummy or a brownie, and that ends up in the paper. There's emergency room visit by a 4-year-old, which is not good. Obviously, this is unpleasant for everybody, including the poor kid probably, but and the parents. But thankfully that hasn't sort of turned into an epidemic of emergency room visits. What I guess has surprised me is the fact that the kind of half-hearted nature, maybe I'm getting political right away, the half-hearted nature of legalization. So Justin Trudeau, who's the prime minister currently and whose policy, election platform included legalization, followed through on an election promise to legalize cannabis. And that's I think been a excellent step forward. But it was kind of like, once that was done, he kind of had expended, or the party he represents had expended the political capital they wanted to do on that. And a lot of the sort of follow-up stuff that I think still needs to be done, like adjusting the advertising and dosage limits and some of the sort of fine tuning of how people can access cannabis, hasn't really happened. And what that's led to is, like, the illicit market has sort of continued to kind of do whatever it wanted in many ways. So that's one thing. The other thing is, like, the economic potential of cannabis. Like Anandia hired large numbers of PhDs, and we leased labs and renovated them, and hired 60 people or more. And it was really, really hard for us to, like, even get any sort of bank to look at us, including the bank that's related to the federal government's own sort of economic development side. It just seemed like cannabis legalization happened, and then Canada to sort of say, hey, well, let's be quiet about it. We don't want to do anything more because it might embarrass us at the UN, or we don't, it's been hard enough to get this policy through. We don't want to infuriate the voters who don't like that aspect, so we're not going to talk about it anymore. And I just think that, legalization has been a positive, but it could be even more positive. I even think about during COVID, the fact that I think alcohol consumption, and I consume alcohol myself, so I could say this, is that, I think it's in general not a positive during COVID. There's lots of people at home drinking too much. If you go out to bars, which is generally closed now in many parts of Canada, this was leading to events that would be like super spreader problems and things like that. And, cannabis seems to be the right sort of social drug for the COVID pandemic, in the sense that people consume it in smaller groups. It's individuals consuming it just to sort of take the edge off the day, because it's otherwise really stressful time for a large number of people. And yet we still have super restrictive laws around advertising or promotion of cannabis, and it's a free for all for alcohol, almost in Canada. So I feel like, I think in many ways, legalization in Canada has worked, but I just wish it was working better, and it was being pushed along a little faster.
I have some more questions around that, but first, because you just mentioned COVID, and we're talking about cannabis, and you're an expert on the biology side of this, I have heard things come up multiple times throughout the course of this pandemic. I haven't heard it in a while. I was hearing it more in the spring and the summer, but I have heard people say things like cannabis can help protect you against COVID. CBD can help prevent COVID. What can you tell us about cannabis and cannabinoids and COVID? Is there anything going on there? And is there any misinformation that you've heard come out?
Yeah, I've done some reading. I mean, there was sort of midsummer, I think I was starting to see stories about this. And a lot of it was preclinical evidence. So not in humans and not in the real world, but more in rodents or other models where, in particular, cannabidiol or CBD, which does have an anti-inflammatory effect, so it can reduce inflammation. And COVID, at least in people who become seriously ill, you often have this kind of overreaction of the immune system. There's this kind of inflammatory response, which cytokines are these signaling agents in our cells that really leads to the body almost mounting too strong a defense to the virus.
That's what they call a cytokine storm.
Yeah. And that's what, people who end up in ICUs may be suffering from. And so the idea is that, and some of the preclinical evidence suggested that CBD could, through its anti-inflammatory effects and some of its, I guess, effects on immune cells, could modulate some, cytokine production or this turn down cytokine storm. But my read on that was that it was more like the entourage effect. There's a plausible hypothesis. Let's do some more work. Let's start, testing this. And it hasn't, as far as I can tell, hasn't resulted in, say, a clinical trial that has shown that consuming CBD helps COVID patients. And I guess, some would argue, well, maybe there's a real world experiment happening right now. I mean, CBD is everywhere. There's lots of COVID. Maybe we could be looking at people who consume CBD, have COVID, and do they progress to hospitalization, or is their hospital stay reduced or something? I haven't seen any of that evidence coming out, but I would say at this point, I'm a bit sort of not skeptical, but just like, well, show us some data, show us some evidence that this is actually real.
So going back to questions around legalization, you've seen the birth of a legal industry at the federal level in Canada. You've got your scientific background and you've got your business background. So you understand the biology and the stuff that's adjacent to that. You understand stuff about how to operate a business and what helps that happen from a regulatory standpoint. Let's say that you were called to a meeting with US regulators and they say, Dr. Page, we would love to talk to you about legalization in the United States. Washington has decided that we're going to legalize cannabis in the US. As a scientific expert and as a business owner and entrepreneur, what would you advise us to do and to not do as we implement this in the US?
Yeah. Okay. So I think there's a few things. One is that I think it's natural with any consumer product, or whether it's a food or alcohol or even tobacco, is that federal regulation should focus on safety. I think there's been surprisingly few cases of illness attributed to cannabis. I mean, certainly people can consume way too much and green out or feel pretty sick, but like things like food poisoning, for example, does not occur with cannabis products in high number. But we do know EVALI, which is this vitamin E acetate vape issue that hit the cannabis industry, but also the nicotine vaping industry last year, is an example where if things aren't carefully regulated, you can harm a lot of people. I mean, I think EVALI killed more than 50 people, right?
Yeah, I think so.
63 maybe. So this would be a failure of regulations. I think EVALI is some of the strongest impetus for federal legalization in the US. But I would say, what I would tell federal regulators in the US is implementing a strong requirement for testing and product safety around cannabis is paramount. That would be sort of a foundational aspect.
Related to that, well, related to that, the issue of testing and safety, I think a lot of people in the US would point to with that vaping crisis that most, if not all of those products were actually from the unregulated market. And so you've got this issue where cannabis is state legal in certain places in the US and it's federally legal in Canada, but you still have a thriving illicit market. And so why is that? Are there regulations that are allowing the illicit market to continue to thrive? How would we combat that?
And are you talking... So yeah, in Canada, the illicit market continues to thrive. And I guess, some of it's historic. It's just like supply chains have existed and consumers have been relying on those. There's also an issue of taxation. If you don't pay taxes, maybe some people are paying taxes at some point in the supply chain, but in general, your dealer is not adding taxes to your bill. So things are cheaper in the black market. You're right in the sense that the EVALI crisis mainly hit illicit vapes, and therefore federal regulations wouldn't impact them. In fact, they might put more sort of barriers around licit production, raise costs, and then therefore lead more people to go to illicit. And I guess that would be another cornerstone of what I would suggest is that I think there needs to be a time where the regulations are focused on safety, but not necessarily on sort of like a high return in terms of taxation, where there's a sort of a period when prices are somewhat kept intentionally lower in order for the regulated market to compete with illicit market, with the hopes that people will eventually gradually migrate to the regulated market. And I know public health, policy people would say that the tension there is if you have low prices, it does encourage consumption. People might be consuming more cannabis than is potentially healthy for them, or it might be more accessible to more people, say younger people who have less money or whatever, and that you're leading to, if you looked at a tobacco kind of model, higher taxes are there to, at least in Canada where we tax cigarettes at a high rate, in order to sort of limit consumption. And so there's this kind of teeter back and forth, push and pull on that. But I guess the other thing I would say is that, my company was bought by a large corporate cannabis player, and I worked as an executive in that company. So I'm certainly not against large companies being involved in the cannabis space. But I would also say, in terms of where federal legalization should go in the US, is that there's the best ecosystem is a mix of the sort of small, medium, and large players, that you have room for the mom and pops. And I have visited, Humboldt, California, and talked to the old school guys. And I certainly grew up in Canada with neighbors who had small grow ups that they had. And those people are part of the economy too. And so I would, and maybe this is sort of some of my sort of lefty viewpoint, but I would hope that, there's room for everybody, and that the playing field is kind of leveled in a way, that regulations aren't so stringent that you need to have like a team of lawyers to get through everything. That there's still some little guys. And, I look at the big players in the US, many who are listed on the US Stock Exchange, and Aurora is a big player globally, and Canopy and stuff. And I think there's room for those big players alongside of small ones as well.
What Are there any new innovations or new cannabinoids that you see coming in 2021? Or are there any cannabinoids that you're excited about besides THC that you think might have interesting psychoactive effects or something like this? I'm hearing more about CBG. I'm hearing rumors about THCV and appetite suppression and even psychoactivity. What do you think is on the horizon with new cannabinoids?
Yeah, I mean the story with those non-THC, non-CBD cannabinoids has been sort of building for a few years now, where the pharmacology of THCV is really unusual. It binds the CB1 receptor like THC, but it's kind of biphasic, I think. So, and it appeared to be having potential appetite suppressing activity at that receptor. So, and then CBG is, showing up. It's present in some cultivars now at relatively high levels, 3% in some cases, 2.5 or 3%. I'm not sure the sort of in vitro pharmacology says as much about CBG's activity as THCV, but I would say both of those cannabinoids will likely start to be emphasized in the marketplace, where there are consumers who are looking for some novelty, producers who are looking for differentiation might start to put out, hopefully it's flower products. I'm not sure I would really be super keen on a CBG vape at this point, not knowing as much. We don't have that much information around that, but some flower products that are, there's these varin strains, these kind of THCV dominant, not dominant, but at least strains where THCV is more abundant, and they would be interesting to see in the marketplace and start to see in terms of feedback from people what they're experiencing. There are these highly potent rare cannabinoids that are showing up in the literature that are not, they are found in the plant. These would be like side chain variants. Like I forget if it's a heptyl side chain.
That sounds right. 7.
Yeah,
and
7.
It's CBT. Is it CBT?
Yeah.
Yeah.
And it seems to be highly potent in terms of the in vitro pharmacology, and I guess it'd be interesting to see if labs start analyzing. I know some labs are doing this now, but more labs would start looking for these other variants and seeing if they are related to some sort of user-reported effect. I'm a little bit. I think we also have to be a bit cautious in the sense that, I'm more confident when it's a flower product with a diversity of chemicals that sort of come from the kind of, maybe it deviates a little bit from the norm of what the chemical composition of standard strains are, but I'm a little more uncomfortable with the sort of THCV distillate. When the idea that we still haven't fully studied some of these more rare cannabinoids and their effects on humans, that they would end up in the marketplace, potentially, high levels of some of them, when we're still sort of scratching our heads around what the effects might be. One of the compounds that we had, which was a CB1 antagonist, this was a purely synthetic product, rimonabant. This was this this blocker of CB1 activity, and the idea was that it was going to be marketed or was produced by Sanofi Aventis. This goes back a decade or something, or more, as an anti-obesity drug or anti-diabetes drug, because it would, if activation of CB1 receptor would trigger essentially the munchies, and people were consuming more, the inverse would be true too. If you block that receptor with a drug, maybe people wouldn't want to consume any food. And I think it worked. They didn't want to consume any food, but they also had no pleasure in anything in their life, in the sense that there was like suicidal ideation, which means, I guess, it's thinking about suicide, and people had, depression, and just, it was not a good drug. It was pulled from the market for this reason. And if THCV, I don't in any way speculate that it has that activity, but the fact that it, appetite suppression goes hand in hand with this sort of blockage of CB1, I would just, sort of counsel caution with some of the sort of in the wild pharmacology experiments that might happen.
Well, how about a question around like the speed of innovation in cannabis? So let's just say, someone decided we're going to make a THCV dominant strain, or we want to make a 1-to-1 THCV strain. How easy or difficult is that? How long would that take, assuming everything was lined up to even try?
Yeah, GW Pharma has published on some of those, a breeding program that took them quite a long time to produce, CBG dominant, THCV dominant, CBC dominant. So it's possible. I'm going to say it could take start to finish, several years to get to these kind of, maybe not 1-to-1. You might be able to move faster because there's some genetics that are already in the sort of gene pool that are showing significant levels of propylcannabinoids. So it's going to take a few years of traditional breeding. It might be faster if you can find the right starting materials. There might be some sort of mutations that are just kind of lingering around at low levels that people could discover.
And we've gone for quite a while now. I do want to ask you about one more area. So you've seen the birth and the maturity of the cannabis industry in Canada and in the US. And now we've kind of got this new, the new kid on the block is the psychedelics industry. And so given your business background, but also your botany and plant chemistry background, what do you make of this industry? How much of it is hype? How much of it is legitimate? And what are you excited about that you're seeing there?
Yeah. So to be truthful, I had been so wrapped up in the cannabis industry that I had very little time to sort of look outward into, and it is kind of like, I sort of put my head up one day and I was like, whoa, there's a lot going on. I've been fascinated with psilocybin mushrooms and psilocybin for a long time as well. I mean, I find just that whole botanical side really interesting. And Tim Hughes and I actually considered doing a psilocybin mushroom genome just right on the heels of the cannabis genome. We didn't get around to it. And
has that ever been done?
Yeah. People have sequenced the genome and they've actually identified the psilocybin sort of cluster, the biosynthesis cluster, or at least many of the enzymes involved in making psilocybin. So I'm going to say I'm very supportive of this change, this openness to psychedelic medicine. I think it's, fully, 60 years or something since like some of the early LSD experiments around addiction and in psychiatry. I mean, some of them actually done in Saskatchewan by sort of pioneers in that area. And so I think there's a huge amount of potential, let's call it healing, but or benefit to humans by consuming these drugs. I mean, Canada were allowing people to consume psilocybin in palliative care situations when they're facing their last days, and people are obviously very upset by where they are in their lives when that's happening, and that this is showing promise. And there's been this ministerial level exemption of people in order to consume psilocybin in a controlled setting with psychologists or psychiatrists. So I think that's really promising. I think there's a lot of other drugs other than psilocybin that are, ketamine seems to be have a lot of value, in depression, and this, a lot of, you tweet about this, so I see a lot of it through your Twitter, the neuroplasticity related to psychedelics. So I kind of view this like an enthusiastic bystander at this point. At the same time, I'm kind of like, I wouldn't say troubled, but I'm a little bit skeptical around this sort of shroom boom, as I've heard it referred to. That, and I think it comes a lot from the cannabis boom, the fact that cannabis generated a lot of wealth for people who got in early. And there's a lot of companies competing in the space. And I actually don't see psychedelics turning into the same type of products as cannabis, which is widely available and used more on a daily basis. Mm-hmm. I know there are people who say, well, microdosing is a daily basis, and there's lots of people who are benefiting from microdosing. But I kind of actually think I wish there was sort of more focus on stronger sort of clinical support and exploration of the science, rather than just a lot of investor money going into sort of Me Too companies that are all seeming to do this. But at the same time, maybe this kind of full bore capitalist approach, as long as it doesn't get, too out of control, is going to, more people studying something can often lead to advances more quickly, just because you have more chance of sort of stumbling on something really important. So maybe that's a benefit. And also the fact that some people are looking at ecstasy or MDMA. Some people are looking at psilocybin. There's ketamine. Is there like an ibogaine sort of thing going on?
Oh yeah. So ibogaine, for those who don't know, is a super potent alkaloid that has very strong psychoactive effects. There's some reason to believe that it might help with addiction treatment, but it also has severe side effects. Like when you take this, apparently you can be tripping in a fairly intense, maybe even terrifying way for like a day or more. Right. So it's very potent. Someone recently synthesized an ibogaine analog, an ibogaine-like drug that had some of the good aspects without the bad aspects. And there's an animal study that indicated that you could make that version of ibogaine.
Right. Yeah. Ibogaine is from this West African tree, or the roots of a tree, and it's used in these kind of sort of coming of age ceremonies. And it must be fairly terrifying to consume this thing at a coming of age ceremony. But, ketamine is it's used as an anesthetic commonly in emergency rooms in Canada, at least. So I just think there's a lot of potential there. And, in general, I feel, I'm a believer in the fact that humans seek access to chemicals that change our state of consciousness. And the fact that, a positive of this psychedelic boom that's happening right now is also the fact that, states and cities are voting to legalize access. We're finally sort of pulling back on these kind of war on drugs controls on things that, essentially psilocybin mushrooms grow. They're not in my backyard right now in January, but, I was at UBC one time, University of British Columbia, and I was meeting with the curator of the mycological collection, and we were talking about psilocybin mushrooms because we both had an interest in these. This is only like 3 or 4 years ago. And I said, oh, have you seen any psilocybe cyanescens, which is these ones with the wavy caps? So they're the locally the most potent psilocybin mushroom.
That's what I've heard. They're more potent than the regular cubensis strain that people probably have seen.
Right. Yeah. So quite a bit more potent. So I said to him, have you seen any cyanescens around? Because they used to grow in like bark mulch under rhododendrons and things on the campus quite a bit. And he said, oh yeah, I'll show you. So we walked out of where we met, and then around the corner, and there's like at the university there's like a sort of an open plaza where there's like the university bookstore, and there's a cafe, and there's kind of this meeting area. And right in front of the Starbucks in the bookstore is this bark mulch bed with these newly planted kind of azaleas or something, and it was all cyanessence. And there's like, there's probably 1,000 students walking by staring at their cell phones, and there's like psilocybin mushrooms staring them in the face if they look. But so, I mean, even that, the fact that these are wild organisms and yet they're banned by government law seemed always to me fairly crazy. So, I mean, long-winded way of saying I'm really supportive of this psychedelic movement, and it just seems to be growing leaps and bounds. But I'm kind of I'm not an investor in any psychedelic companies, and I think I'd be careful about that at this point. Not to say never, but I'd just be really careful.
Any final thoughts you have to share? Maybe even something around, I don't know, some advice that you might give to graduate students or postdocs today who are in the science world, but they might, maybe they have some ideas, maybe they have a vague notion of doing something else or turning the hardcore science into a company or something. What might you say to those individuals given your experience?
Yeah. So I guess what I would say is that at that point, in your life, you need to build a team of sort of advisors and experts, actually at any point in your life when you're starting something new, that you're not going to know how to do it right off the bat. And you sort of have to humbly go to people who have other expertise and ask them for guidance and sort of, build relationships with people who can advise you. Because I just think that it's too much to ask, a grad student with an idea, if it can be, a business success, they need that sort of mentorship. So maybe that's the point. Look for mentors, plural, who can assist you. So I think that's one. The other is that it's kind of it seems like sort of self-help book kind of stuff, but you can't be afraid of failure. Most startup companies fail, and if you go in there really worried about the fact that if your startup idea doesn't work out, that you're kind of you've lost years, or you're never going to succeed in something else. Like you can't have that mindset. It has to be sort of like you're fully committed, but you're also committed to kind of being resilient if it doesn't work out, and it's a little bit like science, right? I always used to counsel people who are new to sort of wet lab biochemistry that most of the time it doesn't work. And building this kind of like mindset of resilience in grad students who are doing that kind of science is really important, because it can, otherwise they just don't make it, right? They drop out because it's too stressful. And so the same thing goes sort of with the startup world. You got to be ready for the setbacks and maybe the complete failures. I would also say the message that I would give is, like, I started Anandia when I was 44 years old, right? I was very, very far from that kind of like grad student postdoc sort of point where people always think that's where startup success comes from. And once in a while, I'll see this on Twitter where someone will have done some analysis, and it's actually like 45 is the average age of a successful startup founder. So I guess the other point that I would say, to grad students or postdocs is finish your PhD or whatever, your master's or whatever or finish your undergraduate degree. I think that's way more important. The timing of it, maybe it's kind of like I'm giving conflicting advice, because at one point I said timing is everything, and Anandia was the right place at the right time. Canada 2013 was the right time to form that company. And if I had waited to finish something, maybe the timing wouldn't have been as good. But I do think I benefited from completing my PhD, completing those postdocs, going to Munich and other parts of Germany, working in the lab, grinding it out, building up that expertise, learning how things worked, all the time sort of learning something new and pushing something. And then when I started the company, I was ready to go. So I would, I'm actually kind of disheartened when I hear people are, 3/4 of the way through their PhD and they're like leaving to do a synthetic biology startup. Like, just get the thing done. Mm-hmm. Like, I'd prefer to see the PhD behind your name and co-founder or founder of a company than just founder. So that would be some of my advice. I guess the other part is that, one thing about the difference between science and startup or business is in science, the currency is, it's publications, it's new knowledge. You're creating something that's what society is paying you to do, right? Could be vaccines for COVID, or could be psilocybin mushroom genomes, or whatever. But what you're doing in business is making money. I mean, it really is. That's the core principle of business. And I mean, there's altruism, and there's corporate social responsibility, and there's being a good corporate citizen and all that kind of stuff. But that's really, the mindset is a big switch. And if you felt the passion for science, because you're creating new knowledge and you're at the cutting edge of something, and you're switching over to the startup world, even if you still have that passion for science, your investors are talking about money. And you have to know that and embrace it and believe in it, or you're going to sort of feel like, what am I doing here? So I think people need that fundamental understanding of that. And, maybe that's obvious, but to someone like me who didn't do business before, that was a shift.
Mm-hmm. Well, John, thank you for your time. New record for me. This is the longest recording I've done so far. I really appreciate everything that you had to share with us. And I think a lot of people are going to really love listening.
Great. Well, it's wonderful to talk with you, Nick. I know we've, over the years, chatted about indicas and sativas and terpene composition. And, in some ways, maybe we could dig into some of that in the future. But it was excellent to chat with you. And the time just went right by. So I have no regrets.